website. He's currently the CEO of Amoeba Energy Company Limited, and in addition, he's a professor at Keio University in a number of graduate programs. And as he also has a position as a fellow of the Earth Life Science Institute at the Tokyo Institute of Technology. He got his PhD in 2004 at Kobe University, and then did a number of research scientist positions at some of the best places in Japan, Riken, the Riken Advanced Science Institute, and Tokyo Institute of Technology. And so he became an associate principal investigator there, and then a fellow that he currently holds. And then in 2017, he became an associate professor in Keio University. And in 2018 is when he established his, I guess you would call it a biotech company. So he'll be talking today. He didn't give me a title, but he recently published a paper entitled "The Remarkable Problem-Solving Ability of a Unicellular Amoeboid Organism and Its Mechanism." And so this is a really exciting developing area. And so to introduce this to you, a lot of this group that attends these meetings is very interested in quantum type biology. And so this is one of these very useful applications of this for very difficult problems. So as you know, computers today are very fast, but they're not very good at solving non-deterministic polynomial problems. And the classic one of these is known as the traveling salesman problem, or TSP. And so in the TSP, the traveling salesman is told by his boss that he has to go and visit clients in every city, and he can only do it once, and his boss is very cheap and says, "I want you to do it the most, least expensive way." So he has to figure out how to do this. This is a map showing the path that would be taken, the least expensive path to travel to every major city in the United States. It took a computer several years to do, and it still is just an approximation. And so the reason for this is that it's an optimization problem, that you get millions and millions of answers, and then you have a regular computer has to solve each answer separately and then go back and rank them in order of optimality. So the difficulty of this problem increases with the number of cities, and because each city, if it's fully connected, each city has all the roads connecting to all the other cities. So as a result, every time you add a city, there's a factorial increase in the number of possible answers that need to be optimized. So I will present some primitive stuff compared to what we're going to hear today that I worked on as a side gig for a while, that I got some DARPA funding. So this came, what is this useful for? Well, a few years ago, Hurricane Rita came and hit Houston, and it was difficult to predict the path of the hurricane. So you only had 48 hours to make a computation to how do you get people out of the way of the hurricane. And so here is the path that the hurricane entered the city, and these are the possible roads. Of course, there's a lot of secondary roads as well that would not be able to hold as much traffic, but there was no plan. And so everybody got on the same road. Everybody ran out of gas. There was no gas, and people couldn't get out of the way in time as the hurricane hit. So in fact, if you're, I think Amazon has made its way to Europe now, and with the pandemic, everybody's ordering lots of stuff off of Amazon. So every morning when the orders come in, then the trucks need to go out and make deliveries to everybody's home, and it's different every day. So you can imagine that Amazon is calculating the most efficient path to make these deliveries because to save on fuel. And this is a supply chain problem that affects all the major stores worldwide. Whenever someone goes into a Walmart, for example, and they check out, then as the item skews on the checkout counter, then that information goes all the way back to the Walmart headquarters and says, oh, this item was just purchased, we need to replace it. So these are done for many, many different important uses, and yet our traditional computers that run on electricity are really not capable of doing a very good job of this at all. So I, with the DARPA funding, got money to build a computer out of DNA molecules, because you have the basic principle of the computation is that you assign different sequences to cities, and you have a sequence that will connect the cities. This would be your path between the cities, and that would hybridize to say that this is the path. So you can optimize the path by having the cities present in numbers of molecules by a saturating amount, like you would for an enzyme kind of reaction, and then you would limit the pathway. So you would have a lot for an expressway, and you would have a smaller amount for a dirt road. And then you would be able to, supposedly, by assembling these, every time you do this, this is a computation. And so you're making a massively powerful parallel computer by having thousands of computations going on at the same time in your test tube. So what we did was to solve a 15-city problem, and at the time it was the biggest problem ever solved by molecular computing. We did a very simple thing where there was going to be an obvious answer, and we start out with so in math speak, then instead of calling them cities, you'd call them nodes. Instead of calling them pathways or roads, you'd call them arcs. But it's, so we added saturating amounts here, and then the optimal answer, we added 100-fold times more of this answer, where it would go in alphabetical order, A to B, C to D, etc. And then these were the one-fold of each of the other suboptimal answers. The idea was, can we make this even work? So in fact, then we mix them all together, let them hybridize, and we had special start and end cities sequences so that we could pick those out. And then it goes ahead and it actually calculates the answer in a very short time. It's just figuring out what the answer is. So you have to, first of all, so this happens very quickly, but then you have to spend days and days sorting this out. And you have to make sure that every city is visited once and only once. If you have something like this where some of the cities were missed, that's an incorrect answer. Or if some of the cities were hit twice, then that's an incorrect answer. So you've got to pull all of these out. Once you get down here, this subset where they're all the correct, but then you have to sort out which is the optimal answer. So this is a lot of running things on gels to separate by length. You get the correct length, where if one is too short, then you're missing. You have double. You have to go through and pull each of these out one by one with DNA sequences that attach to magnetic beads. And then you have to go and figure out what the answer is. And again, we have a probe that tests for each city. So this would be the A-B link, and then we would repeat for the BC and all different combinations, finally run them out on a gel. And so this would be for city D linked to city A, B, C, D, etc. And then quantitate that. And with that, then you can see that A, B, B, C, C, D, etc. came out to be the correct answer. Well, this was tedious. We sped this up by inventing a new qPCR probe that would have the qPCR built right in, and it would test each of these combinations so that we could run all of them at the same time. And we solved an 8-city problem for this. And of course, it looks like a football stadium, where going around the edge of the stadium here is the fastest way, and much faster than running all the way down to the field and up to the other side. So, and we could run all of these at the same time, but even so, this was still a lot slower than what can be done by doing an approximation with a massively powerful regular computer. There is a $1 million prize out there for someone who can speed this up to a point where you can make this kind of molecular computing as fast as a regular computer. Well, the challenge is to have an NP complete problem be solved as fast as a simple math problem that has only one correct answer. So I'm really excited to hear what Masashi has to say today, because he's done this with amoebas, and his research interests have to do with, for example, life, mind, intelligence, computation, information. And so you can imagine that if you're, Alan Turing brought up the concept of what is a computer, and a computer has to have memory. Well, what is that memory? Our memory sits as the answers in a freezer of a combination of DNA sequences. What is the memory here, and what is the origin of memory, and how does this work to solve problems, these complex problems at speed? So with that, Masashi, I turn it over to you, and I'm really excited to hear what you have to say.
Thank you very much for great interaction. So can I share the screen? Is this okay?
There you go.
Yeah, okay. So let me see. Okay. So today I'd like to talk about a kind of biocomputer which uses this amoeba. The name of this organism is Physarum polycepharum, the true slime mold. And then as Wayne introduced, this amoeba will try to solve this traveling salesman problem, which is one of the most popular combinatorial optimization problems. And basically, the reason why I had a chance to, I could have chance to give a talk is that Philip Killian sent me an email, and he was interested in our paper published in 2018, which I'm going to talk about later. And he has maybe some insights that this system can be interpreted as a platform for quantum biocomputing. So to be honest, I'm not quite sure about those quantum physics, but yeah, I'm happy if we could have some productive discussion at the end. Okay, so I think I don't need to explain about my profile, because Wayne have already introduced you. But these are my research subjects I have been involved. So I have now basically theory. I was a theorist to model the complex systems. Then I moved to experiments, which uses this real amoeba as a computing substrate. Then from around 2013 to 2017, in Earth Life Science Institute, I studied the origins of life, which was also a main topic of this session, I guess. And then from around 2018, I established a company called Amoeba Energy. This is not actually the biocomputing company, but we have now tried to develop some electric circuit-based new computer, which was inspired by this the problem-solving mechanism of the amoeba. Okay, then these are the topics for today's my talk. First, I would like to introduce briefly about this amoeboid organism, Physaon polycephalon, and then I'm going to talk about the biocomputer, which I call amoeba-based problem solver. Then briefly introduced how we are now developing bio-inspired computer, which uses the concept, the mechanism of the problem solving of the amoeba. Then finally, maybe this is most important part, but I want to have a discussion with you about the potential application of this system as a platform for quantum biocomputing. Okay, so this is actually the amoeba. So the name is Plasmodium of true slime mold, Physaon polycephalon. And first, I would please have a look at this scale bar. This is 1 centimeter, but nevertheless, this is a single cell, a huge single cell. But it contains many cell nuclei. So if you, so this is a movie I wrote from National Geographic. And this is fast forward movie, but you see in the forest, this true slime mold survives very actively, and you see it's formed this kind of network-like structure and then moves around the space. So in some sense, this organism is a kind of animal, but also it's of some common property of the plants. And yeah, so one of the interesting point is that this organism is a model organism of the decentralized system. The reason is because this organism has a kind of homogeneous architecture. So although this is a single cell, but it contains numerous cell nuclei. So if you cut some piece around here, for example, this part can survive as another individual. So this picture was taken after I put 1, 2, 3, 4, 5, 6, 7, 8 pieces of the part of the single cell. Then they start to spread. Then once they contact, the 8 cells becomes fused into one single cell. So in some sense, it's so yeah, that's the reason why this system is called decentralized. There's no central system, but and also the architecture itself is very homogeneous. Nevertheless, the total system itself can somehow optimize its behavior to respond to environmental stimuli. And this is a life cycle of Physarum. And the yellow stage, which is called plasmodium. So this yellow state, like I showed you the movie, these organisms can make a locomote. And so it's something like an animal. But and also this plasmodium likes humid and dark condition. So basically they like to be under very humid and dark condition without the light. But once they are exposed to UV and also in a dry condition, this yellow color turns to black, and we can observe some sporulation. So this is the figure. Then when one spore opens, one single cell with one cell nuclei appears. This is called amoeba actually. Then many amoeba fuse into one single cell and become and forms plasmodium. Okay, then please have a look at this movie. So this movie was taken by using normal optical microscopy, and you are now watching a kind of Y-shaped structure, and then you can see some fluid flowing from the bottom to the top. This is the protoplasm. And it's very interesting that this organism has some oscillatory behavior. So the period of oscillation is about 1 minute. So you see the direction of the flow reverses around here. And now the flow is, this protoplasm is flowing from the bottom to the top, but maybe like 20 or 30 seconds later, the flow stops and the direction reverses. Can you see the flow of the fluid here? Yeah, so yeah, you see it stopped and the direction reverses. So this kind of oscillation is not, is autonomous, meaning that without any external stimulation, this oscillation keeps, continues by itself. So this work done by Professor Takamatsu in 2000, she created a kind of living coupled oscillator system using a single amoeba here. So this blue part is a kind of barrier, the plastic barrier, which the amoeba cannot overcome. So you are now, this yellow part is a single cell. And this blue and red pixel represent that the thickness of the amoeba decreased and increased respectively. So you can see these two oscillators are performing a kind of antiphasic synchronization. And she regarded this system as a living coupled oscillator system because the length of the channel, the width of the channel corresponds to the coupling strength, and the length of the channel corresponds to the time delay in the coupling. And when the number of oscillator increases to 3, we can observe more interesting oscillation modes. So this mode, which I call 2-1 phase synchronization, is that these two oscillators at the bottom perform the in-phase synchronization, whereas this one on the top performs antiphasic synchronization. And this one, the other one here, rotation mode. In this mode, the phase difference among these three oscillators are 120, right? So it's kind of rotation. And this one is called double frequency mode. In this mode, while these two oscillators at the bottom performs one cycle of the oscillation, this one on the top performs 2 cycles of the oscillation. And these movies are, repeating the same time window. So it's not actually, accurate. So what is more interesting is that we can observe in a real situation, this kind of oscillation mode switches spontaneously without applying any external perturbation. So that means that this system has some intrinsic fluctuation, or in other words, basically the behavior is just probabilistic. Yeah, and then this work is very famous, performed by Professor Nakagaki in 2000. He showed that this organism can solve a kind of shortest path problem. So this black part is a kind of barrier where the amoeba cannot overcome. And this yellow part is a single cell. And once you put some food source at the entrance and exit of the maze, after like 5 or 6 hours later, this organism change its shape and connect the shortest path between these two food sources. So it's a very, it attracted many public attentions and inspired many works after that. Then I'm also, I think I should introduce this work. This work is talking about microtubules in this Pfizer. But the point I have to mention that actually the official name amoeba corresponds to this phase, number 6, whereas the phase we are I'm gonna talk today is in Plasmodium, the phase number 1. So it's the size is quite different. You see, this is a micrometer scale, whereas in a plasmodium, we are talking about millimeter or even centimeter scale. So the spatial and temporal scales are pretty different. But in this amoeba phase, this work shows that microtubules work as a kind of information transmitting channel. So this is the case where you see this is the network of the microtubules. And this part is the source of the chemicals and kind of attractive food. Then this amoeba is able to make right decision to move toward this food source. No, but once you inject some microtubule disrupting reagent, the name is nocodazole. I'm not quite familiar with this field, but you see the network of the microtubules are disrupted. Then the capability of this amoeba to make right decision is somehow disrupted. So meaning that this experiment shows that microtubules are doing something related to making the right decision on at least for chemotaxis. Then you see this is the case where the normal non-disrupted organism individual makes right decision. So you see this is the place where there's the food. Then because of this microtubules, this organism at this stage are comparing two directions, whether to go to the left top or right bottom. Then this here at this moment, this amoeba made a right decision to move to the left top. So this is how they use this microtubule to make a right decision. And so, and also, like I told you, there's some interesting oscillatory behavior. So this is the cartoon of the single cell. You see the direction of the protoplasm flow reverses at the period of about 1 minute. And also, this is the spatial temporal plot, which I took from my experimental system, where this vertical axis represents a kind of area of the pseudopod of the amoeba. And you see, there are some several 10-minute time scale oscillatory behavior here. So there are several different time scales and also the spatial scales. Then let me introduce about our biocomputers. So first we tried to create a kind of neural network computer using this single amoeba. And you see this is what we call a chip or barrier where the amoeba can overcome. And in this chip, there are 16 lanes, each of which we call is kind of neuron. And you see, we assign the state for each of the lane like this. For example, if this lane I is not completely occupied, it's empty, then state 0 is assigned. If it's fully occupied, the state is 1. So the proportion of the area occupied by the amoeba's branch is encoded into a real number in the interval from 0 to 1. Okay, then like I said, this organism doesn't like light. So when we expose some light, this organism wants to withdraw its branch. So we can lead the state 1 by illuminating, we can induce this state 1 to change into 0 by turning the light on here. Okay, so we created this kind of feedback system where we use PC projector that we use for presentation, and then illuminate arbitrary part by changing the image to be projected. And then amoeba, so the amoeba will change its shape where illuminated part will withdraw. Then we monitor the length or the area of each of the branches, then update the illumination pattern here, right? So this is a kind of feedback system. So by changing the illumination pattern, the shape of the amoeba changes. Then the illumination pattern changes. Yeah, and so this is the photo we took for so this is an example of the barriers. And you see this is a barrier put on an agar plate, and this is a single cell. Then after putting the organism at the center of the chip, this organism wants to expand its branches or wants to maximize its body area so that it can maximize the nutrient absorption from the agar plate. So these photos were taken without illumination. So basically the organism wants to expand fully inside the chip. But like I said, we introduced this kind of optical feedback system. So this is a projector and this is a camera. And then this amoeba and chip is put in this agar plate. And we can illuminate the targeted lanes with a very high contrast, like this. Yeah, this is the optical apparatus. So, and then we measure the length of each of the branches using some application outside the system, then determine where to illuminate. So the problem we are trying to solve with this system is a traveling salesman problem, which Wayne has already explained. But yeah, the salesman needs to visit every city just for once and should minimize the traveling cost by choosing the shortest tour among the cities. And this problem is called NP-hard. So one of the most popular combinatorial optimization problems. And the combinatorial explosion occurs. So in case of this figure, so there are 8 cities. If there are 8 cities, there are in total 2,520 tours. But when the number of cities increase to 10, 100,000 tours, then you see if the number of cities is greater than 30, it's hard for even for a supercomputer to find the exact shortest tour, even spending for 10 billions of years. So this is called combinatorial of explosion. And this is how we create a kind of initial state of our Amoeba-based computer. So you see these part. In this system, there are 64 lanes. And in this condition, we are illuminating all of these 64 lanes with the light. So that's why this amoeba cannot enter into the lanes, you see. And by doing this, we are now trying to create initial all zero state. Because each of the kind of neuron, the state is measured as the proportion of the area occupied by the amoeba's branch. By doing this, we can create initial state with all zero. Okay, so this shows that the amoeba actually does not like light. So the light, amoeba does not enter into the illuminated region actually. And also the red and blue pixel represents that the thickness increased or decreased respectively. So this movie is fast forward movie. Actually, the timing for red and blue pixel changes is around 1 minute because it's just oscillatory period. Okay, then I think I should not go into the detail of this kind of equations, but this is what this external feedback system does. So basically, this system measure the length of each of the branch. You see, xbk is the state of each unit, and where a V stands for the city name, and K is the visiting order. So these so in case we have 8 cities, we need to have 64 lanes in this chip. And this is a kind of a neural network, a recurrent neural network, where Hopfield-Tank-Amarie neural network, where you see L stands for the light signal is on or off. And it's determined by weighted sum operation here, by collecting the information of all of the neurons. Then this weight matrix is important. This weight matrix is determined by depending on the distance between city V and U. So by changing this part, we can solve arbitrary problem instance with a single system. But this equation is pretty hard to slightly complicated. So let me explain graphically what this optical feedback system does. So let's take an example of the minimal case where we have just four cities. Okay, so in case we have 4 cities, there are 16 lanes here. And let's see, suppose this lane A1, so branch A1 expanded by 0.2, nothing happens. But it goes to 0.5, 1, 2, 3, 4, 5, 6 lanes will be eliminated. The reason is that when you visit city A first, the salesman should not visit the city A again, second, third, and fourth time. That's why these 3 options are blocked. And also, when the salesman is visiting city A, he or she should not visit city B, C, and D at the same time. That's why these 3 lanes are eliminated. So when this rule is applied to all of the 16 lanes, the amoeba can elongate consistent traveling tour by elongating only 4 branches. And this is the graphical explanation to explain what will be the change for how this optical feedback system reflects the information on the distance between the cities. So in this left example, the branch A1 and C2 elongated by 0.5, meaning that the salesman visits A first, then moves to city C. So the length between A and C is just 1. Whereas in this right case, A1 and B2, meaning that the salesman visits A first, then B second. So the length is 4. So this option is longer than the left option. And if these 2 branches elongated further, nothing happens. But something happens when some kind of perturbating guys, in this case D2, this guy is contradicting with B2, And when this guy elongated by 2.289, this B2 is blocked according to that feedback rule. But this is the same comparison. The same branch D2 expands to 289, nothing happens. But the same guy grows to 0.334, then this C2 is eliminated. This is because, in this equation, the information on the distance between city V and U is, if the distance is larger, it makes the summation, this product summation, closer to the threshold. So choosing the shortest route makes the risk of being illuminated smaller. So basically, this system encourages amoeba to find the combination of the branches, which can minimize the risk of being illuminated by their own perturbating movements. Yeah, so and this is the example of the problem solving process for HCT-TSP. So basically, this amoeba wants to maximize its body area by elongating all of these 64 branches. And at the moment, there is no illumination. But after some of the branches exceed some threshold, some of the lanes will be illuminated. And you see, you can see some propagation of the phase wave, like a spiral wave, or yeah, many different interesting oscillator patterns can be observed. And basically, yeah, and you see these white rectangles are the illuminated lanes. So basically, the organism wants to maximize its body area so that it can maximize the food acquisition, but wants to minimize the risk of being illuminated by the light. And the organism, because of the organism has some oscillatory dynamics, it performs a kind of trial and error to find the minimal risk combination of the branches. And now this organism is now trying to find, almost like finding that there are some lanes which are not illuminated. So that those lanes are maybe comfortable for the organism. And you see C1, H2, D3, E4, G5, F6, and A7, B8 are elongated exclusively. And this combination of the 8 branches corresponds to this tour. You see C1 means the city C is visited first, H second, D third. And so, yeah. And so this organism found one of the feasible solution with a length 128. But this is important that the organism found this route, but this is not actually the shortest one, because we set that the shortest route in this problem instance has length 100. So the one found by the amoeba was not the shortest, but in this case, the worst case solution has the length 200, and mean value was set to be around 150. So the length 128 was not the best one, but pretty good one. So this amoeba is not the exact solution solver, but is a approximate solution solver. Then maybe you notice that this system is the amoeba cannot find solve the problem by itself. It's a hybrid system. It's a coupling of amoeba's oscillatory dynamics and also the control dynamics of the neural net. So the control dynamics, which we call bounce back control, bounces back some bad options of the amoeba's branch with the light. And this bounce back control gives a kind of potential landscape like this. And the control principle itself is very simple. If the light is turned off, branch should expand. If the light is turned on, branch should shrink. But as long as the organism follows this principle, it goes to bad solution. It's called local minimum. But like I said, this organism has oscillatory behavior. So sometimes, even the light is on, the amoeba expands when its oscillatory phase is in the expanding phase. And when the light is off, sometimes the organism shrinks. So thanks to this kind of fluctuation, the organism is able to find a better solution. So basically, this kind of coupling is very important. And we came up with a question. If this fluctuation of the amoeba is random, we don't need to use this kind of amoeba. You know? But in the slides, next slides, I'm going to explain that the fluctuation is not random. And because of the amoeba's branches oscillating with some temporal and spatial correlations, this amoeba is able to find a better solution. And I want you to remember that see, because this organism is a single cell, the oscillative behavior at the terminal of the branches are kind of synchronizing. It's not, random. They are temporally correlated, and also, even though this is, millimeter scale, distant branches are correlated. So there are some spatial and temporal correlation. You see, there are some spiral waves and also phase synchronization, desynchronizations are observed. So that's why this system has some spatial and temporally correlated oscillatory dynamics. Okay, yeah. So then we perform this work. We try to compare the effect of the oscillatory dynamics versus the quality of the solution found by the amoeba. So what we did was to evaluate, we performed the time series analysis and classified the spatial temporal oscillative behavior performed in the center part of the organism. So we measure the number of the cluster of the center part. And it is roughly speaking, if the number of cluster is 1, all of the 64 branches are synchronizing in phase. But if the number of clusters is 2, that means the 64 branches are divided into 2 groups, 2 clusters. And so as the number of the cluster increases, meaning that the whole system becomes less coherent. So we compared this kind of, you see the number of border represents the number of clusters. So we compared how this number of border relates to the quality of the solution found by the amoeba by performing like 20 trials of the experiments. And it turns out that if the number of the border was small, the amoeba was able to find a better solution, meaning that the length of the tour found by the amoeba was shorter. So this is one of the insights that maybe high quality solution is sought by dynamics with stronger spatial temporal correlations. So that means that compared to using purely random fluctuations, maybe in this system, we better use spatially and temporally correlated coherent oscillatory dynamics instead. Okay, so next we try to evaluate the kind of problem size scalability by changing the number of cities of the problem instance from 4 to 8. You see, this is the number of all feasible solutions, possible tours. By changing the n from 4 to 8, the number of solution space grows factorially like this. And we set all of these maps to have the shortest route with length 100 and the longest route, worst case route with length 200. And the mean value was set to be around 150. Then this is the movie. So we performed many trials. Can you see? Yeah. So we were interested in how the time required for the amoeba to find a solution changes or grows as a function of the problem size n. So like you see, for smaller scale problem, the amoeba tends to find the solution faster. Yeah, so in this 4 city case, it's almost like solved. 5 city case solved. So with the increase in the problem size, the time required for the amoeba to, the time required for the solution search grows. But we were interested in the function, the time required as a function of n. So this was the result. Interestingly, the time required just linear time required for the organism to find a approximate solution growed only linearly. And this is the quality of the solution found. So like I said, this is not exact solution, the exact shortest route. And this black dots, 1.0 means the average route length of the maps itself. So if the amoeba is randomly choosing one of the feasible solution, the value here is just 1. But always the amoeba found a better solution than the random sampling. So this was experimentally observed result. Then we tried to create the hypothesis of the mechanism, how this amoeba achieves the linear time solution of the approximate solution. So these are the results of the mathematical kind of toy model, which we call Amoeba TSP. And we could reproduce similar results with this mathematical model. And I think I'm not going to the detail about this mathematical model, Amoeba TSP, but the main idea is that, so this model suppose that the protoplasmic flow or the resource supply from the center pot to the branch is always constant. By supposing this constraint, the amoeba tends to expand almost like constantly the branches. Even when the some of the branches are illuminated, even in those from those branches, the outflow of the resource, the protoplasm is accumulated in the center pot and redistributed to the branches where there's no illumination. And this is a kind of a hypothetical model, but even though it's pretty simple, we could reproduce the same result, the similar result. So the number, the time required to find a solution is just linear, and then the quality found by this mathematical model did not degrade, even though the number of cities grows. And then encouraged by these results, we are now trying to create analog electronic version of the amoeba. So you see this part represents the dynamics of the amoeba. You see there is some capacitance, and when the electric current flows from the center, we observe the charge as an analogy of the length of the amoeba pseudopod. And this part is a crossbar, which computes a kind of neural network weighted sum operation. So you see, these are the branches, and the information is sent to this crossbar. And these are the resistances. These resistance represents the weight matrix of the neural network. So if you change the resistance value depending on the information on the distance between the cities, we can use this system to solve arbitrary TSP instances. And actually, in my company, Amoeba Energy, we are trying to create this kind of application-specific integrated circuit, ASIC. And also, this is an additional information, but also we have developed another Amoeba-inspired algorithm for Boolean satisfiability problem called SAT. This is another NP-complete problem. And this is a kind of digital version of the Amoeba TSP. And you see, according to these logical inhibition rules, this analogy of the Amoeba tries to find the configuration where when illumination is turned on, the Amoeba branch is shrinking. And in the absence of the illumination, Amoeba branches are expanding. And once the system goes into this kind of configuration, the state becomes stable, and we can get a solution. In this case, by putting 1, 1, 1, 1, we can satisfy all of these logical constraints. Yeah, and so actually, we are now developing different types of the combinatorial optimization machines. So this one is dedicated to represent Amoeba TSP using analog circuit, and using this digital circuit FPGA, we can implement an algorithm for solving the Boolean satisfiability problem. And also we tried to create a very simple model for the superconductor amoeba, which uses adiabatic quantum flux parametron. And these days, starting from quantum annealer proposed by D-Wave Systems, these are many Japanese companies trying to create or propose new types of the combinatorial optimization machines. So maybe we should find some application where our approaches have some advantages compared to those other types of the machines. Yeah, then I would like to come back to the main topic, which is I want to have a discussion with you with a potential platform, whether we can use this system as a potential platform for quantum biocomputing. So please remember that for the decision-making capacity of the amoeba, microtubules had some important roles. And yeah, so then this is a paper published by the group of Philip Killian. I was introduced this result, and it is very interesting. And it says that the microtubules are able to transmit information on the optical energy. And it says the range of the information transmission is kind of macroscopic. So putting these results, I would like to summarize the kind of fact checked for productive discussions. So these are the source of the important facts to be useful for the discussion. So first, our proposed system, Amoeba-based problem solver, does not demonstrate exponential speed up for finding an exact optimal solution, exact shortest solution, but shows the capability of finding an approximate solution in linear time. And this linear time approximate solution capability could be reproduced by classical circuit or even by classical algorithm without needing no quantum. So this should be understood clearly. Then, and also the time scale of the our experimental system is like from several 10 minutes for problem solving, and also several 1 minute for oscillation. And the spatial scale is around, it's like centimeter. It's so pretty macroscopic system. I mean, this APS is a platform to examine how the amoeba recognizes its surrounding light stimulation environment as a function of its shape to evaluate numerically the performance in making the decision on minimize the risk of being exposed to aversive light. And amoeba's decision-making capability seems to be supported by forming microtubule networks, at least for chemotaxis. So we are not sure whether the same applies for photo avoidance, which we saw in our experimental system. But microtubules itself are shown to function as a channel for transmitting information on light. And the scale is almost like quite macroscopic. So this is my own view as a starting point for the discussion. So our system may be an interesting experimental platform to bridge between micro and macro. So micro is talking about quantum or molecular microtubule and light. And what we saw is, cellular level, individual level, and also what we saw is telling something like, in the capacity of intelligent decision making to minimize the risk of being illuminated by aversive light. So it's interesting if we could bridge between this micro and macro using our experimental system or some similar kinds of experiments. Yes, that's all for my slides. So I'm very happy to accept your question and have a discussion with you. Thank you very much for kind attention.